come to the Golgi body. It is called as the Golgi body, or it is called as the Golgi apparatus. So, or it is called as the Golgi complex. You can say whatever you can say, that Golgi body or Golgi apparatus or Golgi complex. So, this is the structure of the Golgi. So, the Golgi, it was discovered by Camillo Golgi in 1898. So, the first electron microscopic structure of the end of Golgi body look like this. That means it is a densely colored structure that is found in the electron microscope. Look at the structure, and this is the Golgi body, and it was seen in the electron microscope for the first time, and it was densely, means a very black color or very darkly stained, the color of the Golgi body was seen. So that's why it is called as the densely colored structure that was present inside the cell is called as the Golgi apparatus. So the Golgi apparatus, the found in the all the cell except mature RBC, sieve tube, and swarm of bryophyte and pteridophyte. Okay, so that means mature RBC, sieve tube, and the swarm of bryophyte and the pteridophyte, they don't have the endoplasmic reticulum. So in the animal cell, 3 to 7 complex are present. 3 to 7 means this is the 1, this is 2, this is 3, this is 4. That means 3 to 7 complex structure of the Golgi are present, but it is found very large in size in case of the animal. But in the plant, the number is 10 to 20, but the size is very small. So that means the animal, the number is huge, but number is very low, but the size is very huge. But in the plant, the number is very huge, but the size is very small. And that's why in the plant, the Golgi body is called as the dictyosome. So that means what is the dictyosome? It is simply the Golgi body that is present inside the plant is called as the dictyosome. So come to the structure of Golgi body. This is the structure of the Golgi body. The structure contains 3 types of the structure are present. One is called as the cisternae, one is called as the tubule, and another is called as the vesicle. So what is the cisternae, what is the tubule, what is the vesicle? The cisternae means it's like this flat like structure, they are called as the cisternae. The flat long disc like shape, flat disc like shape is called as the cisternae, and its the size is 0.5 to 1 micrometer. What is the micrometer? Simply the 10 to the power minus 6 meter is equal to 1 micrometer. So that means 1 meter by 10 to the power 6 is 1 micrometer. So this is the diameter of the cistern. So that is another is called as the tubule. What is the tubule? And at the periphery there are many structure looks like this, and this is called as the tubule. And another is called as the vesicle. Vesicle, that means this is a vesicle. So that means a structure in which it is surrounded by a layer and it contains something, that is called as the vesicle. So there are the three types of the structure. One is the cisternae, a long like structure. The tubules, a very small, that is present at the periphery, is called as the tubule. And the vesicle, that means from the Golgi the vesicle arises and it contains some food particle. We will know something after this Golgi body study. We will know about the what is the vesicle. So now come to the cisternae. It is very important. Cisternae means, look at the structure. This is the nucleus, this is the ER, and this is the Golgi. The Golgi part, it has two parts. One is called as the convex and another is called as the concave. Means, let's say this is the nucleus, this is the ER, and the Golgi is present, looks like this. Look at the structure. That means one part is called as the convex and one part is called as the concave. You know what is the convex, what is the concave. The pointed end is called as the convex. So that means one part is here, this part is called as the convex and this part is called as the concave. So the convex part is also called as the C-space, while the concave part is also called as the trans-space. That means here it is called as the C, here it is the trans. That means here this part is called as the cis part, and here this all the large structure they are called as the trans part, or it is called as the forming phase. Forming phase, and it is called as the mature phase. Why it is called as the forming, why it is called as the mature? Means if in the child the size it looks like this, but after the adult, of course the size increases. So that means it is the forming phase, it is the mature phase. Here also look like this structure. That means it is a forming phase. That means it is just formed here, but it is the already matured one. So that's why the trans part is called as the mature phase. And always remember that this C space is facing towards nucleus. Towards nucleus. Look at the structure. This phase always looking towards the nucleus. That means it is nearly present at the nucleus in comparison to the trans phase. So that means this phase is very far in comparison to this C space. So this is the C space, this is the trans phase. The C space is the convex, the trans is the concave, and the C is forming phase, and the trans is mature phase, and the C is towards the nucleus, and the trans is away from nucleus. Okay, so they are interconnected. That means the cis and the trans, they are interconnected here. So this is the main structure of the Golgi body. So now come to what is the function of the Golgi body. So that means in the ER, from the ER, the proteins are transferred by forming a vesicle. Here in the smooth endoplasmic reticulum, the lipids. The lipid, here it is the protein. So they are synthesized and transported to the Golgi. So in the Golgi, the C space accept the protein and the lipid. So always important, the C space accept the protein and the lipid and the steroid. Okay. So what is the function of the lipid? Function of the Golgi. That means after accepting, what will we do? So look, let's say this is the protein. Now the protein comes inside the Golgi. Now the Golgi forms the modification of this protein. That means it helps in the modification of protein and lipid. That means let's say this protein, this enters into like this, here inside lipid, here also it enters like this. So after this it modifies this protein and the lipid. So what is the modification? The Golgi body adds a carbohydrate into him, So the process is called as the glycosylation. So what is the glycosylation? That means the addition of the carbohydrate to any molecule. So here in the protein and the lipid, the main function of the Golgi is it adds a structure here also, it is called as the carbohydrate. That means it modifies the protein and the lipid by adding the carbohydrate, and the process is called as the glycosylation. The second is called as the most important protein sorting. The cell sorting. Sorting means, let's suppose this is a cell, this is a nucleus, this is ER, and this is the Golgi. So that means in the ER, the protein and in the smooth endoplasmic reticulum, the lipid are present, lipid are transported into the Golgi, and after that the glycosylation occurs, and after that it decides this protein or the lipid should go to where. That means let's say there are the many organelles, for example the mitochondria and lysosome, and many organelles are present. Let's suppose this protein is for the mitochondria. That means the Golgi decides whether it should go to the mitochondria or not. If it decides it goes to mitochondria, then this structure, that means after the glycosylation, the structure, it also forms the vesicle like this structure. Here this is the vesicle. The vesicle like structure, now it enters into the mitochondria. Let's suppose it decides this protein or the lipid should go to the membrane, then it forms again the vesicle, and this vesicle fuses to the plasma membrane. Looks like this. This is the protein or the lipid that is needed for the plasma membrane, so it fuses like this. Let's suppose it decides this protein or the lipid should go to the lysosome, so again it forms the vesicle. So now this vesicle it fuses to the lysosome, and now lysosome should get its protein or the lipid. So this is called as the protein sorting. That means it decides which protein or the lipid should go to the which organelles. So that's why the Golgi body is also called as the traffic police of the cell. So like that means in the traffic police, like that the traffic police decides which side of the people should go to where. So like that the Golgi also decides which molecule or the protein should go to where. So this is the main function of the Golgi, and along with that it helps in the formation of cell wall by dictyosome. So that means in the plant the Golgi is called as the dictyosome, and the Golgi of the plant or the dictyosome helps in the formation of the cell wall in the plant cell. And another is it helps in the acrosome formation in sperm. So what is the acrosome formation? That means during the fertilization, let's say this is the ova, this is the sperm. So it is some reaction is required for the fertilization, and this reaction is called as the acrosome formation. So the Golgi in the sperm which also helps in the acrosome formation that is needed for the fertilization. So this is the main function of the Golgi, that means accepts the protein and the lipid, and after accepting it modifies by adding the carbohydrate, or the process is called as the glycosylation. Then the cell sorting, that means it decides which protein or the lipid should go to where. The next one is it helps in the formation of the cell wall by the dictyosome, and another is it helps in the acrosome formation in the sperm. Okay, so this is all about the